Biology, at its core, is the story of life on Earth, and for any species to persist across millennia, it must reproduce. In flowering plants, angiosperms, this entire process centers around one highly specialized organ, the flower. Long before a bud even appears on a stem, hormonal and structural changes trigger the plant to differentiate a floral primordium. The blueprint is set. Looking closely, we see two reproductive systems. At the periphery is the andrecium, the stamens serving as the male structure. In the center is the gynoecium, or pistil, consisting of the stigma, style, and ovary, the female structure. To master angiosperm reproduction, we need to trace the parallel development of these two structures, right down to the cellular level. We will begin with the male pathway. At the very tip of the stamen sits the anther, the primary site of pollen production. A young anther's transverse section reveals a bilobed tetragonal architecture. Each corner houses a microsporangium, packed with sporogenous tissue. Outer layers, epidermis, endothecium, and middle layers, protect and release pollen. The innermost tapetum layer actively nourishes the developing grains. Within that center, sporogenous tissue acts as pollen mother cells. Each diploid cell, 2N, undergoes meiosis, a process called microsporogenesis. The result is a microspore tetrad of 4 haploid cells. As these microspores separate and mature into pollen grains, they develop severe biological armor. The outer layer, the exine, is built from sporopollenin, one of the most resistant organic materials known to science. Beneath that tough exterior sits the intine, a thin, continuous inner wall enclosing the living contents. Inside, the mature grain holds 2 cells. The larger vegetative cell acts as a food reserve, while the smaller generative cell floats directly within its cytoplasm. In over 60% of angiosperms, pollen is shed exactly like this. In the remaining species, that generative cell divides once more mitotically, creating 2 male gametes before shedding. Millions of these armored haploid microspores now stand completely ready for deployment as the mature male gametophyte. Now we track the female counterpart. The pistil anchors the system, a stigma landing pad, a style stalk, and a basal bulged ovary. Inside the ovary is the megasporangium, or ovule. This anatropous ovule connects to the placenta via the funicle. Notice the micropyle opening at the apex. Protective integuments surround the inner tissue, known as the nucellus. A single diploid cell in the nucellus differentiates into the megaspore mother cell. It undergoes meiosis, Megasporogenesis, yielding 4 haploid megaspores. Usually 3 degenerate, leaving 1 functional survivor to develop into the female gametophyte. Through mitotic divisions, this cell expands into the embryo sac. 3 cells form antipodals at the chalaza end. The large central cell contains 2 polar nuclei. At the micropylar end, the egg apparatus forms, 2 synergids flanking 1 egg cell. The synergids have thickened cellular structures called the filiform apparatus. Their specific job is acting as chemical guides for the incoming pollen tube. Count them carefully. Because of the 2 polar nuclei sharing the central cell, the mature female gametophyte contains 8 nuclei, but it is strictly a 7-celled structure. It is now completely primed for fertilization. Both gametes are entirely non-motile. Pollination is the mechanical solution to physically transfer the pollen from the anther to the stigma. Plants use wind and water, but many rely on animals. To secure biotic vectors, they offer floral rewards, sometimes taking extreme forms, like the 6-foot-tall Amorphophallus, providing a safe incubator for insects to lay their eggs. However, constant self-pollination leads to genetic weakness or inbreeding depression. To prevent this, plants have evolved numerous outbreeding devices, actively discouraging their own pollen. This means the stigma is not a passive landing pad. Pollen-pistil interaction is a dynamic chemical dialogue. The pistil evaluates the pollen's specific surface proteins and actively decides whether to accept or completely reject it. Human breeders bypass this mechanism through artificial hybridization. By manually removing the anthers, emasculation, and bagging the flower, they ensure only highly specific pollen reaches the stigma. Once accepted, the pollen grain germinates. In this longitudinal section, the pollen tube carves a path through the stigma and style. Guided by the filiform apparatus, it enters the micropyle, penetrates a synergid, and discharges 2 male gametes. The immense physical gap between the male and female gametes has been bridged. The stage is set for a climax unique to flowering plants. Here, the sequence splits into two distinct fusion events. First is syngamy. One haploid male gamete fuses with the haploid egg cell. This forms the diploid zygote, which will eventually grow into the embryo. Simultaneously, the second male gamete travels to the center and fuses with the two polar nuclei. 3 haploid nuclei merge to form the triploid primary endosperm nucleus. This is known as triple fusion. Because these two distinct fusion events, syngamy and triple fusion, occur within the exact same embryo sac, the phenomenon is termed double fertilization. Biology dictates a strict order of operations here. The endosperm tissue must develop before the embryo. This guarantees a ready food supply for the growing zygote. Yet, biology also allows for exceptions. Some species bypass this highly choreographed sequence through apomixis. It is a form of asexual reproduction that mimics the sexual process, yielding fully formed seeds without any actual fertilization taking place. We also see variations like polyembryony, where multiple embryos develop within a single seed, a common occurrence in citrus fruits. Tracing these pathways, from the first division of a mother cell to the precise mechanics of double fertilization, maps the foundational architecture of the seed. And it is this exact biological mechanism that sustains global agriculture, and by extension, all of human survival.